Thanks for coming to this session today. It's nice to see you all. As David said, my name's Angie Peltier. I work out of the Crookston Regional Extension Office, and I'm going to report on 4 projects. I'm going to rely on David to, again, give me the hook. Please let me know when we have about 5 minutes left. I'd like to refer you guys also to this research book. If you didn't pick one up when you came in the front doors, make sure you pick one up on the way out, because it's going to contain information about a couple of the projects. So you can look at the treatments and everything in more detail in this book. Oh, for crying out. So there's 4 different projects, and what I'm going to do is introduce them one at a time. And as we go through each one, you'll notice it's not me alone. There's an incredible team behind each of these projects, and so they're listed on the title slides as well. Again, all of these projects were sponsored by Minnesota Soybean and Research Promotion Council, either in full or in part. So the first topic that I'm going to talk about is a new way of managing white mold in soybean. So this research was done both at the Crookston Regional or Northwest Research and Outreach Center, and then down at the Central Lakes College Ag and Energy Center in Staples. Crookston, we had 22-inch rows. Staples, we had 30-inch rows. So as you know, white mold can affect both yield and quality in soybeans. We consider it a disease of high yield potential soybeans. So a lot of the things that we do to try and maximize our yield potential, so early planting, narrowing up row spacing, high plant populations, all of those things tend to increase our disease risk. We do have several means of control. But because we don't have the threat of white mold every single year, a lot of the cultural practices that we can use to manage white mold don't necessarily make sense from a maximizing yield potential standpoint. And so we really suggest that people continue to maximize for high yield potential soybeans. But if you do have a lot of disease potential in your field, there's many things you can do. Partially resistant varieties is one. Rotating away from some of our broadleaf crops, so many are susceptible to white mold, that can help, although it's not a cure-all. Foliar fungicides also can provide some control, but they're not perfect. It's been theorized that we have some coverage issues. Foliar fungicides tend to work as protectants, and so we need them to be at the site of infection before the spores land. And so the site of infection is typically at leaf axils where flowers are starting to die, lower down in the canopy. And so having that fungicide penetrate the canopy and getting good coverage there is key. And so that's what this project was aiming to do, was to try and improve our fungicide coverage down and within the canopy. One thing that you might be wondering, what side dressing nitrogen on the corn has to do with white mold management? Well, side dressing nitrogen into corn requires a specialized implement. So a lot of people that do this apply nitrogen, kind of spoon feed the crop during the growing season. They use something called a Y drop, and it dribbles liquid nitrogen towards both, rides down within the canopy, the corn canopy, and it dribbles nitrogen towards the root line in both rows. But what this allows for our purposes is there's a newer product that attaches a nozzle body down within the canopy. And we figured, well, you know, maybe applying over the canopy isn't the key here. Maybe we can improve coverage by applying down within the canopy. And so the commercial product is this 360 undercover. They're very expensive, and so Jeff Nielsen at the Northwest Research and Outreach Center took out his welder and used some chemical resistant hose, some skid plates from that folks use so that they can easily move through the field when harvesting sugar beets. And what he did was he built a spray boom. And so we have both the normal spray nozzles that can spray the fungicide up and above the canopy, and then we have units that can ride down within the canopy and between rows. So here's what this looks like in 30-inch rows. So the white circle is showing us the nozzle that's riding above, and then down below you'll see with the black circle that's our nozzle body riding down within the canopy. So we have 3 nozzles there. 2 are facing either row. Flat fans just basically spraying up and down, trying to improve that coverage within the canopy. And then we have a third that kind of sits at a 45-degree angle to spray up under the canopy. So we tried to keep everything the same except droplet size and where those nozzles were positioned. So sometimes chemical manufacturers will recommend that folks try and get coarse or very coarse droplet sizes, just so that those droplets have enough mass to move their way down and within the canopy, don't just float away. We want them to be at the site of infection. Everything else, though, was the same. We had the same carrier volume, so 10 gallons per acre. 5 mile per hour application speed. Pressure was the same, 40 PSI. And you can see here for the over-the-top application, at these pressure and at the pressure and speed here, this particular nozzle was able to provide very coarse droplets. And for the within the canopy, we had a different nozzle, and that provided medium-sized droplets. So what we did, I had a picture somewhere of this actual plants, but I couldn't find it anywhere. So what we did is pounded into rows some nice rebar fencing that we cut off. And to that, we actually attach these little alligator clips. And then to those alligator clips, we attach this water-sensitive paper. So the water-sensitive paper starts out yellow, and it moves to blue when moisture hits it. We let it dry after application, stuck it in labeled Ziploc bags, and brought it back to the office, where we used a dedicated scanner to scan in those water-sensitive papers. And then we used a free program called Deposit Scan that was developed by a USDA. It's free for anybody, so if you have an interest in it, let me know. I can give you that information. But we use that to then objectively look at fungicide coverage. So here's what we found. So this is results from 3 years, 2020 through 2022. And what we're looking at here is the results in 30-inch rows. So we did have 2 different spray booms because we wanted those between the rows within the canopy to kind of ride down the center between rows. So here, this is in 30-inch rows. This is percent coverage. The 2 bars on the left, that's at 6 inches above the soil line. And the 2 bars on the right, that's 12 inches above the soil line. The red bars, that's the over-the-top fungicide application, and then the yellow bars, that's the within the canopy application. You can see on the right-hand or left-hand side here, at 6 inches above the soil line, we more than doubled our fungicide coverage when we applied down within the canopy. We didn't see the same thing, although we did see the same trend, but statistics tells us there's just too much variability. We didn't find statistical differences in at 12 inches above the soil line. Here's in Crookston, so this is in the 22-inch rows, same setup here. So on the left, that's 6 inches above the soil line. On the right, that's 12 inches above the soil line. Red over the top application, and then the gold is within the canopy. Again, at 6 inches above the soil line, we more than doubled our fungicide coverage. We saw the same numerical trend at 12 inches above the soil line, but again, too much variability. Statistics tells us that those aren't, real differences. It's probably just due to chance. So, big question: why is coverage improved at 6 inches and not 12? So my running theory is, you think about soybean plants as they progress through the growing season, some of those lower leaves that are no longer receiving as much sunlight, they begin to drop from the plant. So it's more open. The canopy is much more open down towards the lower end than the middle of the canopy. And so it's my theory that maybe some of the leaves that are there did interfere with coverage at the stem. I'm willing to entertain other theories as well. So here's another project I'm going to report on. So this is a IPM survey. I do see Oliver sitting in the back of the room. He was a scout a couple years ago. He now works for a seed company in North Dakota. So this is in addition to providing data that's of value to farmers. We want to make sure that folks have a heads up if we have a major pest outbreak. This survey was in part, it helped us to figure out that we had the pyrethroid resistant aphids several years back, and so it's a valuable survey. I am referring you to pages 20 through 26 in this on-farm research book if you want more details. This past year, we had a heck of a time finding interns. If you know somebody that is interested in being a scout and getting training in both wheat and soybean scouting, let us know. We're willing to pay students handsomely to work with us and get that training. Okay, so a couple highlights from 2022. The main thing we saw was some grasshoppers showing up. So the green triangles here show those locations that were scouted in June that had between 1 and 25 nymphs per square yard. So as far as spray thresholds are concerned, when we start getting concerned is when we have 50 to 75 nymphs per square yard in the field margin. Here's what we saw for adults. We did get up to 6 to 10 adults per square yard in field margins at some of the locations, and this is in July. But for adults, the spray threshold is 21 to 40 adults per square yard in the field margins. And so we didn't reach thresholds. I know that grasshoppers did reach threshold populations in some wheat fields, especially around Crookston, but not so, at least in the scouted fields in soybean. So we did see a little bit more soybean aphid than we've seen in recent years. So the red triangles, that's anywhere from 80 to 100% of the plants that are scouted in each field. 31 plants are looked at. So those are the plants that had aphids. What do the populations look like? Well, they didn't get so high. We only had them go up to 101 to 200 aphids per plant on average. And as you all know, the threshold is 250 aphids per plant on average. Spider mite presence, I'm just going to compare and contrast 2022 to 2021. So here, all of those black dots, those are scouted fields where they didn't find spider mites on the edge of fields. Here's 2021. So very different. Obviously, 2021 was a drought year. 2022, a lot wetter. So that's the kind of difference a year can make. Here's our in-field spider mite presence in 2022. Again, just didn't find them. Here's where we were at middle of August 2021. So again, night and day. So again, there's plenty of additional information, pages 20 to 26 in the on-farm cropping trials booklet. So here, we're talking about a program we call Minnesota SCN Squad. So you might recall a couple years ago, I presented on a project where we were trying to get a 30,000-foot view of what was happening as far as SCN population densities in fields, especially up in the Northwest where we were, you know, this was the area of the state that was most recently infested. So during that year, we had 32 events just in the Northwest. I personally spoke to over 1,800 farmers. We distributed 5,400 sample bags, and we only had about 6.7% submitted. You'll recall recently we've had several really wet falls where, you know, there's other things that take precedent, you know, not wanting to harvest soybeans in snow. Understandable. So farmers have other priorities, but we still want that 30,000-foot view. We kind of need to know where we are, just so that we're able to communicate that with folks and get people just more interested in actively managing SCN. So that's where this project comes in. The goal here was to hopefully interest the next generation that doesn't have as many pressing tasks, hopefully during harvest time. Interest the next generation in soybean cyst nematode, careers in agriculture, and soybean research in general. And so we call this program Team Soybean. We have nice little stylized soybean cyst nematodes here looking very angry, and then we have the superhero Team Soybean. So this program was marketed to FFA chapters, select FFA chapters. So we really targeted those in the Northwest again, and then in the Central Lakes and dairy regions as well. So we had 19 FFA chapters participating. This was we were able to offer it to 42. We didn't have 42 takers, but you'll see the northernmost were up in Polk County. We had, I think, I can't recall the exact FFA chapters, but if you want more information, give me a call. But we went all the way down. We had one chapter down here in Chatfield, but so many in this Central Lakes region as well. So here's how the program worked. So the goal was to so I'm not going to get to my last topic at all. So the goal here was to make it as easy on the chapter advisor, the teacher, as possible. So basically, the program would pay for a sampling kit, a nice sample probe, bucket, basically everything they would need in order to collect samples, sample bags, even pays for shipping those samples into the lab. So we also provided PowerPoints to that teacher. So introducing the students to the pathogen, why it's important, its effects on soybean, and what we can do about it. The students then take turns, either taking samples on their own home farms or on the farms of volunteer farmers, collecting those samples. The samples are shipped to the lab for analysis. We receive the raw data, Extension does, and then what we do is we've created two separate sets of data, one for the teacher as sort of an answer sheet, and one for the students that just contains the raw data. The teachers has also raw data in addition to management recommendations. So then the students interpret the data, report results to farmers. We've also created fill-in-the-blank press releases because people in the community want to know where their checkoff dollars are going. They also want to know what's going on with SCN in their area. So then students, they submit this press release into the local paper. Win-win-win for everybody. Farmers wouldn't necessarily have to collect their own samples, but they could get the data, and the students would learn valuable skills that they would need as they pursue work eventually, hopefully in agriculture, because we sure need people in agriculture. So here's where we are in the process. Late this week, we are going to be sending the data out. Took a while to get all of it in, but and actually we've had some people just send in their results this week, or their samples this week too. So we're still kind of in that process. But the main goal after we complete this process is to also have sort of a career panel where people that work in extension, people that work in industry can talk to students about their own experiences, their own jobs, and what they entail. And because it's just so important for kids to see themselves in these positions going forward in order to motivate them to stay in agriculture. Okay. Last one, I'll just introduce it and the sponsors and the locations, David. So this one is detailed in your book, pages 6 through 8. We call it Planting Green Along the Red, pages 6 through 8 in this book. And this is looking at a cereal rye winter cover crop, planting soybeans green into that cereal rye. And then what we're doing for these on-farm trials is we are varying when that cover crop is terminated, 1 to 2 weeks prior to, at, or 1 to 2 weeks after soybean planting. Okay, so we have a lot of people involved here. Quite a few sponsors. Primary sponsors, Minnesota Soybean Research and Promotion Council. And here are the on-farm research locations. Just so you know, this fall, 2022, so these are the 2021 sites that were planted to cereal rye in the fall. We had one in the Northwest. We had a couple of locations bow out. And then down in West Central Minnesota as well. We do have 3 locations that were planted in 2022 to cereal rye in the fall, around the Crookston Research and Outreach Center, and then 3 around the West Central Research and Outreach Center.